Prepare it to live.
Ready? Yeah. I've admitted everyone. Hello, everyone. Welcome to the first webinar by the Center for Brain and Mind. I'm very excited to welcome all of you to the first in this webinar series. Today, we'll be discussing genetic risk factors of psychiatric disorders and modeling that risk via cellular models. We will put a schedule of speakers for today in the chat shortly. You mute. From eLife, who would be speaking about the new peer review model at their journal, as well as on the Ben Barres Spotlight Award program. I'm very fortunate to be one of the awardees of this program last year, and we've recently set up the 3D cortical organoid model in the lab, and this award has made it possible for me to go to labs abroad and seek the cutting-edge technologies which we can set up in our own lab to probe this model further to establish the cellular basis of psychiatric disorders. Just a couple quick logistical things. At any time, if you have questions for any of the speakers, please post them in the chat box. We'll take it up at the end of their presentations. And I will now turn it over to Professor Reddy, who's the CBM lead investigator at NIMHANS and head of the psychiatry department, for his opening remarks.
Professor Reddy, you're muted.
Good evening, everyone. It's my great pleasure to start this program, Center for Brain and Mind webinar, first of its series, titled Unveiling Minds, Genetic Insights and Cellular Mechanisms. Center for Brain and Mind, supported by generous funding from Rohini Nilekani Philanthropies, has it functions from NIMHANS and also for NCBS Bangalore. Myself, I'm the leader. I'm the chief, principal investigator of the program from NIMHANS side, and here Dr. Professor Raghu Padinjat, who will speak later on from the NCBS. Now, prior to this Center for Brain and Mind, we had the ADBS program, which was supported by the Department of Biotechnology and Pratiksha Foundation. And the main purpose of this particular research, when we started up 5 years ago, was to really look at the shared and distinct pathophysiological and genetic mechanisms of serious mental illnesses. We decided to study things with a cross-cutting using 5 serious mental illnesses. We decided to study schizophrenia, bipolar disorder, Alzheimer's dementia, alcohol dependence, and obsessive compulsive disorders are all highly familial disorders. We actually decided to study loaded families, that is, probands with more than one affected first-degree relative. We have made a substantial progress, and we are in the process of following up not only the index probands, but also their affected and unaffected relatives. And Center for Brain and Mind is, one sense, a continuation of this ADBS program, but also has many other objectives, which I am sure Professor Raghu Padinjat will elaborate later on. It's a very huge and ambitious program with a lot of people working on this program, both at NCBS and at NIMHANS. We are today joined by Dr. Stuart King from eLife, Professor Kristen, Brennand, the Elizabeth Mason House Jameson Professor of Psychiatry and Professor of Genetics at Yale University School of Medicine, and Ole Andreassen, Professor in Psychiatry at University of Oslo, Norway. Of course, we also have Dr. Raghu Padinjat, who will be talking today. I welcome all the participants, and I wish the webinar a great success. I'm sure this webinar will lead to a series of more intellectually invigorating webinars in the near future. Thank you, and I don't want to hold up for a long time. I think Dr. Stuart King is already waiting to begin his first lecture. Thank you, Ritika and Bizu.
Thank you, sir. I'd like to now invite our first speaker, Dr. Stuart King, who is the Research Culture Manager at eLife. He'll be speaking to us about the new peer review model at eLife, as well as give us more information about the Ben Barres Spotlight Award. Stuart?
Thank you very much. So yeah, hello everyone. I am Stuart King, and it is my genuine pleasure to be here today as part of this event tackling such important questions with potential for real impact in people's lives. To have the chance to talk with you about, as Dr. Ritika said, how eLife's initiatives and new publishing model can help accelerate scientific discovery. So let's start with a little more about eLife. So eLife was established in 2012 as an independent, not-for-profit publisher, supported by funders and led by editors who, like many of you, are all active scientists. eLife's mission is to bring about real change in how research is reviewed and communicated, and we work towards this mission in 3 essential ways: through the open access journal eLife, which you may have heard of, through the development of open source technology, and also through active community engagement. Earlier this year, so back in the end of January, we transitioned to our new publishing model, which focuses solely on providing expert public review and assessment of preprints, which I will talk more about later. So, as already mentioned, the reason I'm here today is because Dr. Ritika Sud was recognized by eLife with a Ben Barres Spotlight Award last year, which was awarded based upon her reviewed preprint that had at that point been evaluated by the organization PeerRef, then has since been published in the journal Disease Models and Mechanisms. And thanks to the funds from that award, Dr. Ritika has now had the opportunity, as she explained much better than I have, to travel to the US to learn cutting-edge techniques from experts in the field. And that's something that I'm looking forward to hearing how that goes. The awards themselves, for a bit more context, are awarded from eLife with the aim of providing both funds and greater visibility to researchers from underrepresented backgrounds and countries with limited research funding. The awards themselves are fairly flexible, and they are judged based on the potential of the grant itself to unlock new opportunities for the winner, their research, and their wider community. Importantly, and kind of as evidence, the awards themselves are open not only to people who have already published in eLife, but also authors who have reviewed preprints with publicly available reviews from other organizations, as we've already shown. And at the moment, the next round of awards will be opening next year in 2024. So eLife runs these awards in memory of our remarkable colleague, Ben Barres, who served as a reviewing editor for eLife for a number of years before sadly passing away from cancer. The awards themselves are a tribute to Ben's passionate championing for greater equality and equity in science, as well as his fearless commitment to challenging the status quo. And then along the kind of theme of changing things and challenging the kind of dogma in certain areas. So eLife's new model is itself an attempt to rechange how peer publishing and peer review can and should be done. So to give some more detail, eLife's new model is designed to provide expert public review and assessment of articles that have first been made available as preprints. So preprints that we receive that are submitted to us are then sent to peer review if the editors are confident that they can find reviewers to be able to produce high quality reviews that will be valuable for the research community. So that's the criteria we're using to make the decision about what we peer review. Importantly, and perhaps where we're most different from other journals, is that we no longer issue an accept reject decision after that peer review has taken place. Instead, everything that we review, every preprint that eLife reviews, becomes published on the eLife website as a reviewed preprint, and then that is published alongside a eLife assessment that summarizes the strengths and weaknesses of that preprint, as well as the public reviews from the reviewers, and if provided, the response from the authors as well. So all of this is made transparent. The authors then have the kind of power to choose what they do next. They can choose if and how they want to respond to the reviewers' comments, including in that author response. It could be making revisions, it might not be, but it's up to them, it's their choice, it's their work. And at any point after the publication of that reviewed preprint, once the authors are happy with it as a piece of work with their, associated with their name, they can choose to have it be made the full version of record, which is the version that ends up being indexed and would count towards things like a more traditional publication. So this is how that looks in practice. So this is a reviewed preprint from the eLife website, and you'll see that immediately below the abstracts, in a very prominent place, there is this eLife assessment that will communicate the editors and reviewers' assessment of both the potential impact and the quality, the strength of evidence of this piece of research, using a common vocabulary that is intentionally written to be both kind of neutral and understandable by a broad audience, people who are interested in research. We also have sort of other indications to let people know that this was a preprint that's been reviewed by eLife, a breakdown of the time frame over which this takes, and it's sort of highlighted that it's generally faster process than traditional publishing, and access to the full peer reviews are also prominently there. So the reason we've adapted this new model is because we were looking for a way to basically improve and kind of eliminate some of the barriers that are experienced in more traditional journal publishing. And we kind of would argue the benefits of the new model in a number of ways that I'll just go through now. So firstly, it is faster. So reviewed preprints reduce the delay from submission to publication from around 9 months, which is what we were seeing kind of in our process last year, to around 2 to 3 months, which is what we're seeing this year with the new model. Plus, by also removing that accept reject decisions, it's eliminating those kind of all too common but sometimes unhelpful cycles of rejection, revision, and resubmission. Secondly, it's good for science because by providing these reviews, making them open, we'll be giving a very rich and nuanced assessment from experts in the field about a specific piece of work, so that readers have access to it and they are able to better understand the strengths and weaknesses of that work in much more detail than anything you would ever be able to get from just using just the journal name or potentially the impact factor to try to understand. Simultaneously, the transparency of this work being made open through a preprint and the reviews being made open too means that there's greater scrutiny of the process, which again is a good thing for science. For scientists, this approach still offers a published output that can be submitted for hiring and grant applications. So like I said, it's the author's choice to make their work turned into a full version of record, which would still count for these purposes. But they also have the added benefit of this short, accessible summary of that work that really highlights why this work stands out, what's good about this work, and why it's important. That can also be useful for aiding those decisions around hiring and funding. And really, the idea here is it's really shifting the focus from the evaluation being about where you've published, which shouldn't be the way it is, to really focusing on what you've published, which is obviously much better. Like I've said, with kind of more elements of the process being made public and also being made public sooner, the new model brings a lot of transparency over more traditional models previously at eLife and elsewhere. And I think that kind of the big things is you can see how a piece of work was assessed, because that is also made transparent. Kind of the fifth point here is around that the new process is really giving authors much more control. A lot of controlling the publishing process has kind of been taken over by reviewers, determining what revisions need to be made and what barriers and hurdles authors need to jump over to get their work published. We think that authors are a better judge of what makes their work, what their work should be, and what they're happy to have published under their name. So we've given more control back to authors. So like I said, in the new model that we have, it's the author's choice when their reviewed preprint is finalized and when they're happy for it to be the full version of record. They can choose how to respond. Like I said, they could address things in comments rather than making changes, argue why the changes that the reviewers have requested aren't appropriate for their particular work. And again, they may even choose not to make them. They may choose to just address them in that letter. And sort of the last point, so because this process is actually much more efficient than a lot of other journal processes, there have been some savings that we have then passed on back to authors. So like I said, eLife is a not-for-profit. Our fees cover our costs, and what we've been able to do because of the savings is reduce our publication fee from previously $3,000 down to $2,000, which we charge at the point which we commit to peer reviewing the work. Like I said, this is only to cover our costs, and if anyone is experiencing financial barriers that prevent that being something that they could cover, which is understandable for many places and sort of situations, we offer full waivers. That is not an issue. If you request a waiver because you need a waiver, we've never declined one, and it doesn't affect, and that information isn't passed on to editors, so that's not a concern either. So those, like I say, are the key benefits that we've seen from what we believe and have starting to see from the new model, and this is why we believe this is a better way for people to do research. If you would like to learn more about this, I've shared some links and an email address. You can also contact us with questions, or also some social media places you can follow. But of course, I'd be very happy to take your questions now. Thank you for your attention.
Thank you very much, Dr. King. Are there any questions?
The questions may be put on the chat box, please. We'll just give it a minute and then we'll start off the next one. So let's see.
I can help by answering questions that other people have asked before to get the idea.
Yes,
going.
that's a good one.
Yeah.
That's a good one. Yes.
So as I mentioned, we transitioned to this new model back in January, and it's now kind of 6 months on. So we now have more of a kind of understanding of how it's going. And in fact, actually this week, well, it's like 7 months on, but this week we published an update on that, which is again available on the eLife website if people want to read more. What we can let you know is that we've seen since the new model a very steady stream of submissions. We did anticipate that, as with things, if you change something, people want to see how it goes before they decide to submit. So that is ever so slightly lower than what we were having before, but it has been very steady since the start of the year. So we're very pleased about that. And the other thing to say is that the rate at which submissions have been encouraged for peer review is also consistent with what we were seeing before. So a concern that you may have is that editors might be more conservative, given that they, if by making a decision to review something, it could eventually become fully published. So we had anticipated that maybe they would feel more selective, but that's not what we're seeing. It's very similar to what we were seeing before. So that's some updated information that I didn't have on my slides.
Thank you.
Thank you. Okay, I think we will proceed and move on. So it's a great pleasure to invite the next speaker, Professor Raghu Padinjat. So he is the Dean of Research at the National Centre for Biological Sciences in Bangalore, and also the lead investigator at the NCBS for the Centre for Brain and Mind. He's a clinician turned scientist and has a key interest and has been working for the last many years in the field of inositol signaling in human brain diseases and neurobiology. So I invite Professor Raghu Padinjat to give the talk.
Thank you, Biju. Can you hear me?
Yes.
Okay. Good evening, everyone. In the next half an hour or so, what I'll try to do is describe a program that we've been pursuing collaboratively with NIMHANS over the last 5 years. to try and understand a very important group of medical conditions, the neuropsychiatric disorders. So by way of introduction, I'd like to spend a moment thinking about how function emerges in a nervous system. The ultimate output of the brain is behavior, and that is the function of the brain. And this behavior arises as a consequence of the underlying physiology of the neurons and glial cells that make up any nervous system in the animal kingdom. This physiology, in turn, arises from the wiring up of different types of neurons to form circuits, and the function of those circuits, in turn, therefore, depends upon the function of the individual cells that make up a brain. So therefore, to finally understand the functional output of a nervous system, it's important to be able to study the brain at all of these levels of analysis. Now, there are many ways of understanding how the nervous system functions, and that depends entirely on what the question you're asking is. And you can use a series of animal models ranging from the worm C. elegans, which has only 300 or so neurons, all the way through to mice, which have approximately 10 to the power of 8 neurons. All of these systems have their own strengths and weaknesses. In particular, the use of model systems with fewer neural cells allows an easier mechanistic dissection of the underlying processes. But what about studying the human brain, which by 3 log orders of magnitude is more complex than its nearest mammalian counterpart, the mouse brain? So the human brain has about 10 to the power of 11 neurons. The history of studying the human brain using patient studies to try and understand function is very old and very influential. I illustrate on this slide 2 well-known examples. The first is the work of Pierre-Paul Broca back in the 1880s. Broca studied patients with stroke leading to speech loss, and his work on these patients led to the identification of what we now know as the speech motor center. Likewise, in the late 1890s and the early 1900s, the work of Wilder Penfield, a neurosurgeon who stimulated the brain during open neurosurgery, led to the mapping of the homunculus, which is shown on the bottom right-hand side of the slide. Effectively, using such studies, he was able to define parts of the motor cortex that were able to control movement in different parts of the body, be it the lower limb, the upper limb, the face, tongue, and so on. So clearly, such studies can help us understand how the brain is organized and how it functions. And early studies such as these using human patients have formed the foundation of subsequent work that has elaborated our understanding of how a complex brain is organized and how it delivers function. Fast forwarding back to the modern era, I'm going to talk to you today about the program that we've set up here in Bangalore. The goal is to study psychiatric syndromes, and these are defined as illnesses that interfere with the way a person behaves, interacts with others, and functions in normal life. There are a number of disorders included in this, including mood disorders, personality disorders, schizophrenia, substance dependence or addiction, anxiety, and autism spectrum disorders. They are an important problem worldwide. In India, approximately 2 to 3% of the population is at risk. And according to the latest Global Burden of Diseases study, 193 million people in India, that is approximately 14% of the population, are estimated to have some form of mental illness. The most common ones being depression and anxiety, followed by intellectual disability of unknown origin. These diseases have a prolonged course of evolution and hence treatment, and they therefore also have very important social and economic impact. Now, the etiology of psychiatric syndromes has been studied for a long time, and to summarize a very large body of work, it's now well recognized that there is both a genetic component that leads to disease susceptibility. And there are also environmental factors, both prenatal and postnatal, that result in brain plasticity, leading to disease susceptibility. Thus, as per our current understanding, it's a disease that has both genetic and environmental contributions. And the extent to which this happens can result in a person with a brain on a continuum ranging from the healthy at the one end to severely ill patient at the other. Despite a lot of work, there remain a number of important clinical challenges in handling psychiatric disorders. We still struggle to define risk factors and also diagnostic markers that can help us define disease types, as well as the progression of the disease. And in the domain of treatment, we lack the predictive power to say who will respond or not respond to an existing therapy. We do need novel compounds that might help us to treat the diseases, these diseases. And further, there is also the possibility of repurposing existing compounds in different ways to treat the diseases at different stages. Now, all of these challenges could be dealt with if one could understand the basic neuroscience that underlies mental illness. For example, what is the genetic basis of mental illness? What are the neurodevelopmental mechanisms that are altered during the development of the brain in a patient with mental illness? We need to understand how the brain responds to environmental factors and overall understand the cellular and molecular principles that govern this plasticity. In order to do this, one needs to design a study that can capture several of these features. First of all, the study should be able to capture the evolution in brain structure, physiology, and function as a person goes from being well to a state of having a mental illness. Second, we need to be able to identify the underlying genetic and environmental contributions. And finally, one wishes to study the cellular basis of disease development. So in order to do this, NCBS, along with the National Institute of Mental Health and Neurosciences, has embarked on a program to use the clinical resources available at NIMHANS to identify suitable patients in this study. And this is done through the various specialty clinics at NIMHANS, such as the Schizophrenia Clinic, the OCD Clinic, Addiction Center, and so on. Our strategy has been to use what are called clinically dense families. And an example of one such clinically dense family affected by bipolar disorder is shown on the slide. There are two features that are of importance here. First of all, that these families have individuals affected at multiple generations, as is shown on the slide. Second, there are often multiple individuals in a single generation that are affected, along with, for example, this one individual marked in blue who does not have clinically defined illness. Why is this important? Well, the reason for this arises from the genetic and social structure of the Indian population. What's shown on this slide is a measure of what is called heritability by descent in different population groups within India. And as compared to the Ashkenazi Jews, which are shown where my arrow is, there are many populations in India which are several folds higher of IBD compared to the Ashkenazi Jews or the Finnish population. And what this means is that there is a greater chance of discovering homozygous loci that might contribute to severe mental illness in this population. So during this study, we've been working towards recruiting families with such clinically dense features, 250 families affected by schizophrenia, BPAD, OCD, substance dependence, and dementia. In an ideal situation, the goal is to recruit at least four individuals per family, the proband, a second patient, and two unaffected individuals, if possible, one above the median age of risk and the other below the median age of risk for different purposes. The idea then is to characterize both the patient and the controls at multiple levels of brain structure and function, ranging from clinical behavior or phenotype, clinical phenotype or behavior, brain structure as measured by MRI, brain activity as measured by functional MRI and clinical electrophysiology. All of these measures would constitute a putative composite endophenotype, and this would be consisting of several different things: clinical assessments, biochemistry, neuropsychology, electrophysiology, and neuroimaging. And we hope to do this as a serial prospective cohort, assessing individuals and controls at intervals over a 20-year period, and finally create a digital database to facilitate research. Now, all of the studies that I mentioned so far are able to cover, at least in part or in whole, the levels of function in the brain that are now shaded, that is behavior, physiology, and in some cases, circuits. But what it does not allow you to do is to study the function of individual cells in the context of mental illness. And that is, of course, because the brain in a living patient is really not available for biopsies. However, we do need to study these illnesses at the level of cells. And this is because one wishes to understand what are the changes in synapse development, what are the changes in synapse chemistry, for example, neurotransmitter release, neurotransmitter receptors, metabolic changes, and finally, changes in neuronal function in terms of excitability and the electrical response of neurons. So in order to do this, we've undertaken a program to generate a repository of induced pluripotent stem cell lines from each of the patients and their unaffected family members. And the overall workflow for doing this is shown on this slide. One can then take these stem cell lines and differentiate them to give neural tissue in vitro. And the overall workflow here is shown. One starts with neural stem cells, and then over a period of 120 days or so, if you differentiate them, you will see the appearance initially of immature neuron markers, then later mature neuron markers, cerebral cortical markers, finally synaptic markers, and those markers for genes involved in the synthesis of neurotransmitters, and some markers of excitatory neurons such as CAM kinase 2 alpha. And cultures derived from this look like so, with the neuron shown in green and the glia shown in red. One can take such cultures and study them for physiological analysis, as shown on this slide. If I play this video, I hope you can see the fluctuations in fluorescence, both in the cell body as well as in the dendrites of the neurons. And these represent changes in calcium, intracellular calcium levels that are a reflection of ongoing neuronal activity. As the cultures mature in age from 7 days to 45 days, the number of such calcium transients progressively increases, and the amplitude distribution also is able to change. And such studies allow you to monitor the physiological development of the neurons in vitro as a surrogate for what happens to the brain during development in vivo. It's also possible to develop these cultures for much longer periods of time. And shown here is an example of a 70-day in vitro culture. And what you will see here is the synchronous firing of neurons throughout the culture, which is quantified on the panel below. And this is an indication of the development of network activity within these neurons. A final thing that one can do is to monitor physiological development in these cultures. For example, looking at the reversal in GABA activity in the developing brain. Now, all of us are familiar with the role of GABA as an inhibitory neurotransmitter in the brain, the adult brain. However, early in development, GABA is an excitatory neurotransmitter, as was first shown in this paper way back in 1995. And in these cultures derived from human iPSCs, it's possible to capture these stages of evolution. On the top are 3 different cells shown, where the application of GABA results in an elevation of calcium transients, indicating an excitatory role for GABA. And in the bottom 3 panels are shown 3 cells in which the application of GABA results in a silencing of calcium transients, akin to the inhibitory role of GABA that we are familiar with in the adult brain. Lastly, the program is undertaking a set of experiments to try and determine the genetic basis of any altered neural function that one might observe in these cultures or might see in the clinical phenotyping by genome sequencing and analysis. So overall, to summarize what I've said so far, the program has 3 verticals: a prospective cohort, which is clinically phenotyped every 3 years, a workflow that results in the generation of iPSCs that can then be differentiated to study cell biology and cell physiology, and finally, genome analysis that allows us to address the genetic basis of what one might observe either in vitro, in the cultures, or in vivo in the clinical phenotype. For a single family such as the one shown over here, therefore, the program is generating a number of resources. For example, there would be data on the endophenotype, there would be genomic sequence, there might be iPSCs that have been generated, and neural stem cells that have been generated. And therefore, for any individual who wishes to study a particular problem, this set of resources is available for you to design an experiment and carry out your program of discovery biology. It's possible, for example, to understand the difference between control, shown in green, and bipolar patients, shown in orange, or as an alternate case, to compare a drug-resistant and a drug-sensitive person, shown in green, red and green respectively. Up to now, over the last 5 years, the program has carried out an enormous amount of work that is summarized over here, and the resources that have been generated, be it clinical data, genetic resources, or cell lines, is summarized over here. And this is available in the form of a biorepository to anybody who wishes to design a particular project and carry out experiments using these resources. Finally, the program is also developing a research-level database where all of the data from the 3 verticals will be deposited, and you will be able to log in and with suitable approvals, carry out whatever multimodal analysis you would like to do. Then the next few minutes, I'll try and then describe a few projects that have been carried out so far using the different verticals of information that the program has generated. And the first of these is a very important study about the diagnoses and symptoms seen across all of the families that we've collected so far. And this was led by Sriraj. And what the essential conclusions of this study are is that more than half of the families have subjects with different diagnoses within the 5 severe mental illnesses that we're studying. He also noted that a significant proportion of the unaffected participants also have subsyndromal clinical symptoms. And this is very important from the point of view of choosing controls when one is comparing patients with so-called unaffected relatives. And it makes us think about what is the best way to choose controls for these studies. The second study that is led by Saipriya and Srinivas Balchandar looks at the effect of adverse childhood events on neurocognition. And this study has identified that multiple adverse childhood experiences worsen the occurrence of cognitive defects, even in unaffected first-degree relatives of patients from multiplex families. I think that you get the sense that from the large amount of clinical data that has been collected in this program, it is possible to do multiple types of analyses and derive conclusions that can inform on better patient management systems as we go forward into the future. The program has also carried out genetic analysis using the exome sequencing that's been carried out. One example is shown over here, but by no means the only one. And this study looked at the whole exome sequencing of the individuals in these dense families and found that there were a number of variants distributed across the family. And a large proportion of these overlap with Mendelian disorders, genes implicated in already known Mendelian disorders. And this is very valuable because it helps us try and understand what might be the developmental origins of some of these severe mental illnesses. Finally, I present a study in which Salil and colleagues looked at the abnormalities in the migration of neural precursor cells in familial bipolar disorder. And you can see here that there is a difference in the migration pattern of neural precursor cells in controls over here and BPAD patients on the right. And the essential difference is that there is a much more of a random movement pattern in the patient-derived NPSCs compared to wild type. Okay, so after looking at describing some studies that have examined neuropsychiatric syndromes in each of these domains, I want to now turn to what we originally envisaged as the real strength of this program, which is to study mental illness across multiple scales, ranging from clinical symptoms in humans through to structure, functional impact, cellular impact on neural function, and that arises from genetic changes leading to altered protein activity. And as an example, I'll present some work that has been started on studying a family with bipolar disorder. This is a relatively small family shown over here, in which all patients in F1 are affected by BPAD. But the difference here is that the 2 patients, D19 and D20, have very severe mental illness, severe form of the disease, compared to the other members, D23 to D26. And this assessment of severity was done using a number of quantitative features as analyzed by Ravi Nadella and Viju. And based on all of these criteria, 19 and 20 were classified as severe disease, and 23, 24, and 25 as mild to sometimes not a moderate illness. So we wanted to understand what might be the basis of this difference in severity of phenotype. And to do that, we carried out an analysis of the inheritance of damaging variants in the clinically mild and severe cases of BPAD. And this work was done by Shubhra when she was a project assistant in my lab. She carried out an analysis of the damaging variants from the whole exome sequencing of all of the individuals shown here. And she identified a number of different variants that were only inherited by D19 and 20, that is the severe cases, but not D23, 24, and 25. I'll describe to you today, not all of those variants, but just one of them, which is a variant in GRM1, which appeared to be a variant inherited only by the severely ill patients, but none of the mild cases within this family. Now, of course, exome sequencing allows a rapid identification of a large number of variants that could contribute to disease. But how can we establish that these variants are functionally relevant and they do impact neuronal function and lead to aspects of the disease phenotype? So what's GRM1? Well, GRM1 is part of the metabotropic glutamate receptor family. There are 3 groups, and all of them are expressed throughout the nervous system and are activated in different ways. And GRM1, which I'm going to talk about today, is in fact a receptor that is expressed both in the presynaptic and postsynaptic terminals within the human nervous system. It's also expressed in glial cells in the central nervous system. GRM1 and its variants have been studied many times before in brain disorders, and this is a diagram depicting the distribution of GRM1 variants that have been described in schizophrenia, bipolar disorder, and spinocerebellar ataxia. In fact, a homozygous knockout mouse in GRM1 results in spinocerebellar ataxia rather than a primary neuropsychiatric disorder. Anyway, the variant that we identified in this family is S189G. And S189G is present within the extracellular ligand binding region of the metabotropic glutamate receptor. It's very close to the ligand binding site between lobes LB1 and LB2. And after an extensive structural bioinformatics study, including ligand docking and molecular dynamics, Rohini Kristen, who's a postdoc in my lab, reached the conclusion that S189G is likely to be a destabilizing mutation that affects ligand binding and therefore full activation of the glutamate receptor. So to test this idea, we carried out a number of experiments. The first of these was that we simply generated wild type and mutant GRM1, expressed it in HEC293 cells, and studied the response of the cell to activation by DHPG, which is a glutamate agonist. And when you do that, what you find is that the GRM1 S189G, which is shown in orange, shows a lower calcium influx pattern compared to wild type that is shown in blue. This is quantified here at the bottom, and you can see that at different concentrations of DHPG, ranging from 25 to 12 to 6.25, the mutant receptor is always activated to a lower level compared to the wild type. So that's fine. And this kind of heterologous expression suggests that the S189G variant is in fact a loss of function or a partial loss of function mutation. But what about its effect on the neurons in the brain? And to do that, what Shankanil did was to take iPSC lines from D19, which is a patient with severe phenotype, and D25, which is a patient with a mild phenotype, and differentiate them to give a culture which is composed largely of neurons, but also has a small number of glial cells. And you can see the neurons shown over here stained with MAP2 and some proportion of them which express CTIP2. He then loaded them up with a calcium-sensitive dye of 203 and measured the calcium transients at 45 days in vitro. And when you do that, what you see is that both the patient lines, D25, which is mild, and D19, that is severe, have much higher calcium transients compared to D149, which is the control, which is shown in green. Thus, as one might expect in a patient with BPAD, the excitability of these neuronal cells arising from the patient is greater than that in wild type. The second thing which he did was to try and ask the question, what happens when you stimulate cells with DHPG? DHPG is a chemical ligand, and it is used frequently as a surrogate for glutamate in studies of metabotropic glutamate receptor. And so when you stimulate the cells, the forebrain cortical neurons that we derived with DHPG, you can see the response of the control cells, which is in green. We are measuring here intracellular calcium levels, the mild case, which is shown in blue, and the severe case, which is shown in orange. And from measuring hundreds of these cells, one can then plot the average rises in intracellular calcium. And what you see is that the rise in calcium in both the severe and the mild case is lower, with the severe case giving much smaller rises compared to the blue, which is the mild case. The green, of course, is the control, and you can see that over there. As a control, we also stimulated these cultures with NMDA, looking at the activation of a distinct but related class of glutamate receptors. And there you can see that while the activation in both the mild and the severe lines is higher than in wild type, it is no different between the severe and the mild patient. And finally, during KCL depolarization, you can see that the severe case is slightly more excitable than the wild type case, than the mild case. So overall, these kinds of studies that try to bridge the gap between genetics, patient phenotype, and the disease in a dish model have led us to conclude that GRMS189G by in vitro analysis is a partial loss of function allele. Remember that as an SNP, it is present as heterozygous and probably is present in combination with wild type neurons. It depends on whether it's biallelically or monoallelically expressed. It impacts glutamate signaling and excitability in forebrain cortical neurons in a disease in a dish model, and is correlated with this. That is correlated with the severity of BPAD at the clinical phenotype level. And we're currently trying to analyze the data from structural and functional studies in the brain using MRI and EEG to ask whether we are able to bridge the gap further and therefore establish a role of this particular variant in the severity of BPAD in this family. Of course, I would hasten to point out that there are multiple other alleles that are also inherited by the severe cases, but not the mild cases, which almost certainly will contribute to disease phenotype. But this is really an illustration of what one can do with the resources that this program has generated in doing this type of multi-scale analysis. So I'll end here by acknowledging all the people involved in this program at NCBS, STEM and NIMHANS. I think the program has had more than 100 staff up to now who've all worked very hard to make this particular effort worthwhile and success. And I should also thank the patients and their families for agreeing to participate in these studies. The clinical studies in particular are very taxing, and getting patients with mental illness to participate and give so much time to this research is really very noteworthy. I'd also like to thank multiple agencies that have generously supported this program: the Department of Science and Technology, Pratiksha Trust, the India Alliance, Rohini Nilekani Philanthropies, and NCBS. And I'll stop here and take any questions.
Thank you, Raghu. Thank you. It was a great talk. Let's wait for questions. Is there anything on the chat? Give a few minutes. I think there is a question. Let's see.
Where are the questions? I can't actually see.
I'll just tell you. So that's easier. Okay. I think, okay. Can we unmute some people and get the questions?
Yeah.
No, can we, Pravesh, I think we'll just let you ask your question. That's easier. Just unmute.
You want to be able to?
He has to.
Pravesh, do you want to ask?
Sure, I can ask a question. Can you hear me?
Yes, go for it.
Yeah, so I mean, it's a series of questions, but the overall point being simply that the DBNS ADBS sample is relatively tiny compared to all the population scale data sets that are being used world over. So once we start analyzing multiple disorders at multiple scales, across imaging modalities, across cell lines and whatnot, we're always going to be statistically underpowered. So that was my first question there. And with respect to the same point, the related follow-up is, what are we doing within the ADBS DBNS sample to prevent or guard ourselves against overfitting to the sample? Multiple people are analyzing the same data set, and each result is guiding the other studies, but there is no independent sample. It's all being done on the same sample. So at one point, you do run the risk of circularity or double dipping kind of a thing.
Yeah, okay. I'll invite Janardhan and Biju to contribute to that answer. But before they do so, I just want to point out that this is not a study based on which relies on a large sample size where you're trying to associate gene markers with particular phenotypes. If that was the case, your point is well taken that these are highly underpowered for that type of analysis. However, these particular families, in my mind, are in fact useful for studies as individual standalone units. In the case of the family, for example, that I showed you, the phenotype that we study and the variants that we study might in fact be private and is not present in any other family. But the goal here is to use those variants to try and uncover the function of that particular gene in contributing to the development of structure and function in the nervous system. Once you have an insight such as that from a single family, if you want to extend that conclusion to a much broader population, yes, you will need to do a much larger, you'll need to analyze a much larger population looking for variants or phenotypes that are related to it. So I would say that this study is in fact a stepping stone to a subsequent study where you will analyze a much larger population by the more traditional methods that you are familiar with. So now, regarding the double dipping, whatever question that was asked, Janardhan and Biju, would you mind answering it? I'm not well positioned to answer that.
Dr. Reddy, are you there? You can unmute him. So I think the question was, how do we, what procedures are deployed to guard against overfitting? Essentially, there are multiple analyses happening within the same sample. So that's the primary question.
Yeah, I mean, it depends upon what you are looking at. For example, if you look at the primary purpose, how we started collecting the sample, we collected these loaded families with the intention of looking at various shared and unique endophenotypic markers of severe mental illnesses. I think for that purpose, this sample is fairly good enough, but it may not be good enough for the kind of things that Professor is talking about. But for example, if we're looking at, let us say, neuroimaging markers of, let us say, of a particular illness or a particular psychopathology, I think the sample is decent enough to ask those questions, and most endophenotypic markers are much easier to identify in enriched families than non-enriched families. Now, having said that, no sample is enough. I think we are continuing to collect the sample. If I answered the question partially, I don't know, but yes, I mean, there are multiple phenotypic markers here. I mean, we're actually phenotyping the sample using various psychopathological measures. One leading to another research question is not a new thing, but at the same time, we should guard against hair splitting and see things which don't exist, obviously. For example, looking at one of the examples that Raghu was talking about, what is the effect of childhood trauma or abuse on psychopathology? It might lead you to certain findings, and then those findings can be probed again in the same sample, or if you have different measures, But in any case, all these findings have to be obviously externally validated in some other samples. That remains an issue in research in any case. Now, the third one, what are the plans in for the first open access public release of the sample? I think we discussed recently, 1 year after the complete curation of the data sample, that means maybe about 1 year from now, when all the primary objectives of the endophenotyping and other measures are trying to answer all those questions, and I think it will be a open access after that. That's the nearest answer I can get. Bizu, you would like to clarify anything further other than this last question? Maybe you can focus on other two things.
Yes, I think everything has been answered. Thank you so much. I think we'll move on. Dr. Amog B, do you want to answer your question? I mean, ask your question. You can unmute him, please.
Yes, sir. Thank you for a great talk, sir. I just wanted to ask how useful are these iPSC models to study acquired factors in disease? So would certain epigenetic alterations reflect in these models for, say, childhood trauma? Is it useful to take a cohort and develop these models?
That's a difficult question to answer, but I think that it's important to remember that when you reprogram a somatic cell to make an iPSC, for the most part, but variably, you do erase most of the epigenetic marks. So it's unlikely that you'll retain those epigenetic marks and be able to correlate those to the factors that led to their initial establishment. I think that if you want, also, I mean, I think there's a general problem, which is that while there are many clinical studies that have identified environmental factors that feed into the etiology of severe mental illness, the mechanism by which that happens at the cellular and physiological and cellular level is not so clear. Now, whether it's childhood trauma or socioeconomic status or whatever, presumably they lead to some physiological response of the body, and that is translated into an impact on brain function through some messenger system, most likely hormonal. It will be important to study those kinds of biochemical changes that link environmental factors to brain function. And for that, the program has been collecting and storing plasma samples. And depending on what you think is the mediator of environmental factors to brain function, a detailed biochemical analysis of those plasma samples may be valuable in enhancing that type of information, that kind of understanding. I hope that answered your question.
Right. We can take one more question. Dr. Sushil Kumar Joseph, do you want to ask your question? Sushil Kumar.
Hello, I'm audible?
Yes.
So my question is like being optimistic. So all this project, is it designed to develop some sort of objective blood-based diagnostic test to, diagnose mental or psychiatric disorders?
Yeah, I mean, well, I think that, as I mentioned in one of my introductory slides, clinicians, and you have several of them at NIMHANS who are on the call, would love to have better tools that help them to diagnose mental illness, preferably objective ones such as biochemical or genetic markers. It need not just be to diagnose illness, but also perhaps predict the progression of the disease or predict the response to treatment with various drugs that are used for treating brain disorders. I think the value of this particular program is that it, by mechanistic analysis using these samples, you can come up with candidate molecules that could then be further studied in greater detail for development as biomarkers. Just like the answer to the question from the previous member of the audience, it's very unlikely that from studying the 1,000 or so patients in the cohort as it stands right now, we're going to come up with a biomarker that can be employed from next week or next month. But what is certainly the case is that by innovatively employing the tools and resources that are being generated here, it's possible to come up with likely candidates. And then a much larger biomarker study, which typically runs into thousands or tens of thousands of patients, will need to be done to test its validity before it can be extended to clinical services and to patient diagnosis and care. I should also re-emphasize the point that Janardhan made, is that we've selected these clinically dense families to identify various cellular and genetic markers. It's important to verify these on a much broader population, including those who don't have a family history, before one even thinks of applying it to the care of patients on a day-to-day basis. But I think this is a good platform to start, but it is not sufficient to translate into a biomarker.
Perfect. Are there any more questions? If so, this is the time. Okay. I think that is that. So thank you so much, Raghu. And let's move on to the next speaker. Atika.
Thank you, Viju. Our next speaker is Professor Kristen Brennand. Her research combines her expertise in human stem cell models, genomic engineering, and neuroscience to identify the mechanisms that underlie brain development, traits, and disease. Her mission is to unravel the mysteries of human genome within a collaborative, inclusive, and supportive training environment. With that, I'd like to ask her to please share your slides and we can begin.
Hi, it's my pleasure to be here. Let me just get my slides up. So hopefully that's full screen for everybody. Please interrupt if I'm not sharing correctly. But I'm all good. Great. I'm so excited to be here and share some of our work about using stem cells to explore the genetics of brain disease. And so with that, I'm going to dive in. I think all of you are aware that psychiatric disorders are far too common, far too severe, but I do want to emphasize the extent to which we believe they're heritable and genetically complex. So as the first case study example, I'll point out the heritability of schizophrenia, which is amongst the highest of the psychiatric disorders. Twin studies estimate the heritability to be as much as 80%, but we still can't explain all of that heritability. Genome-wide association studies can explain about a third of that known heritability. Rare variant studies, 5% more, but there's still a lot of knowledge that we're missing. If we open up this comparison across the diversity of psychiatric disorders, we can see that some disorders, schizophrenia, autism, ADHD, and bipolar, are all extremely heritable, 75% or more. But at the other end, we've got depression and post-traumatic stress disorder that are perhaps, 30% heritable with a huge environmental component. But whether we're looking at the top end or the bottom end of this plot, across these disorders, we still have, this problem that we can only explain, a quarter or so of the heritability that we know must exist. So diving further into schizophrenia, and I'm using this as an example because the genetics here are very well powered, and we can really see how heterogeneous the genetic risk is. So, the first variants really identified were these copy number variants. These are the green dots. They are highly penetrant. So the odds ratios here are, up to 100. They're rare. Each of these is found in less than 1% of patients. And they're pleiotropic. So they don't specifically necessarily confer risk to schizophrenia, but broadly to neurodevelopmental disorders, autism spectrum disorders, and epilepsy. More recently, these protein truncating variants, the red circles, have been identified in the last year. There's almost a dozen of them now. These are likewise very penetrant and very pleiotropic, but about 10-fold more rare than the copy number variants, which is why it took us longer to find them. At the other end of this plot, we have these common variants, single nucleotide polymorphisms that are extremely common. Each of them occurring, in almost up to half the population, but with very small effect size. We're talking 1% increased risk for schizophrenia or less per single nucleotide polymorphism. And to give it context, each of us, it's estimated, was born with at least a dozen risk variants for schizophrenia, but patients born with, on average 60 or more. These common variants can be plotted like this Manhattan plot on the right. This is the most recent GWAS for schizophrenia. Everything above that horizontal red line is genome-wide significant. And what we're seeing here are these vertical lines that are actually composed of dozens or hundreds of single nucleotide polymorphisms inherited together in linkage disequilibrium plots. And so these GWAS are informative about the structure of schizophrenia, but not necessarily informative about the treatment of schizophrenia. There's a lot of challenges here in this space. First, how do we translate these vertical lines, these loci, to the gene or genes that they're regulating and the pathways that are impacted? How do these hundreds of risk variants interact in some? And finally, how does this impact clinical penetrance and expressivity? Why might two people with the same genetic risk have very different clinical presentations? Now, these are some big questions. And I think, it's important to consider how best to answer them. Mouse models have been tremendously informative in neuroscience. They're really fantastic at revealing the complex interactions of genes, circuits, and behaviors, but all models have limitations. And the limitations of mouse models include the inability to study the impact of non-coding common variants, in large part because they're infrequently conserved between rodents and humans. And second, the difficulties in capturing the complex interactions between variants, again, because it's difficult to engineer and breed dozens of risk factors into the same line. And finally, the challenges interpreting pleiotropy and variable penetrance and expressivity when we're looking at one inbred mouse strain. And I think you'll note these are exactly the types of variants and problems that we need to understand in order to understand complex genetic psychiatric disorders like schizophrenia. We need to be able to look at non-coding common variants and interaction and across backgrounds. And so this is the reason really that we have been using human stem cell models. I think classically, we think about using stem cells to look at the impact of one risk variant or gene on phenotype. But the questions that my lab is asking more recently are actually not direct genotype-phenotype relationships. Yes, of course, we obviously need to understand how a genotype impacts phenotype, but we're actually asking questions more about how to modulate genotype-phenotype relationships. We know there are environments that make phenotypes more severe, where phenotypes are more penetrant and expressive. An obvious example would be a stressful or traumatic environment. Likewise, we assume there are environments where phenotypes are less severe. All of us, I think, would like to achieve our genetic maximum, be as healthy as possible for as long as possible. And how do we change our environment to ensure that each of us, lives our most healthy life? And so I want to also emphasize that when I say environment, I don't necessarily mean the weather outside. I'm talking about the environment around a genetic variant. So we're talking about all of the other genetic variants that this one variant might be inherited with, so donor genetics. We're talking about the cell type, how variants have different effects in different cell types. And finally, the context. So the hormones, the inflammatory status, the other cell types in the neighborhood, everything that's happening around a cell and how that might influence genotype-phenotype relationships. So coming back to schizophrenia, I want to give you a few examples of how we're studying gene environment or gene interactions in a dish. And the first example I want to talk about is a rare variant. It's the copy number variant at 2p16.3. It's actually really unique CNV for a few reasons. One, again, it's pleiotrophic. It confers risk to not just schizophrenia, but autism. Two, it's the only copy number variant to impact a single gene, Neurosciences 1. But don't let that fool you. It's not a simple deletion. This is one of the most highly alternatively spliced genes in the genome. So diving in, again, here I'm showing you that it's linked to both autism and schizophrenia with odds ratios across psychiatric disorders that are quite high. A unique thing about the Neurexin-1 heterozygous phenotypes are that they seem to be human specific. While Neurexin-1 full knockout mice have, severe phenotypes on synaptic function and behavior, the heterozygous mice have no phenotypes, no behavioral changes, and no synaptic deficits. And this is quite unique to what we see in human people and human neurons. And so in this particular example, Thomas Sudov's lab has engineered the same mutation into both mouse and human iPS cells, and then induced both species of cells into glutamatergic neurons using NGN2 induction, and is showing us here that there is a phenotype in the human neurons and not in the mouse neurons when you assess them in the exact same manner. So again, this is a human-specific heterozygous phenotype. Diving in, why might it be that we have a phenotype in human neurons and not mouse neurons? Well, looking at the alternative splicing of Neurexin-1, we can see that it's highly complex. So this is long-read sequencing, start to finish, of individual Neurexin-1 isoforms. Work started by Thomas Sudov's lab and advanced by Peter Schaffel's group, where we're looking at the vastness of the Neurexin-1 repertoire, first in mouse. So these plots are quite complex, that we've got the Neurexin-1 locus from left to right. And if an exon is included in the sequencing, it's colored green. If it's skipped, it's colored white. The most abundant isoform is at the top row. The least abundant isoform is the bottom row. And so from top to bottom, they found in this study 250 splice variants for Neurexin-1. Peter Schaffel's group has gone on to show the complexity of the Neurexin-1 repertoire varies by brain region, with regions that have more cellular complexity also having more Neurexin-1 complexity. And you can see this even within cell types, so we can, distinguish types of neurons based on their Neurexin-1 repertoire. We started by asking the same question, but in human neurons. So is Neurexin-1 spliced so diversely in postmortem human brain and in stem cell derived neurons? And so here what we're showing you is data from 3 adult brains, 3 fetal brains, and stem cell derived neurons. Again, exons that are included are colored, skipped are white, and the most abundant isoforms at the top, the least abundant isoforms at the bottom. And what I've colored in orange are the isoforms that we found both in the brain and the stem cell derived neurons. And so what I want you to take home here is that the most abundantly expressed isoforms are shared between the human brain and stem cell derived neurons, which really gave us the confidence that we could begin to ask questions about how patient derived mutations impact Neurexin-1 splicing in the dish. And so here's our cohort. It's small, but again, these are very rare mutations. I want to point out that deletions in Neurexin-1 are non-recurrent. So they have different boundaries in every individual. We have 2 patients here with heterozygous deletions in the 5' region of the gene, encompassing the promoter and first 2 exons. This is a mother-son pair defined by bipolar disorder with psychosis. And we have 2 with deletions in the 3' region of the gene, the 2nd, 3rd, and 4th from last exons. And this is a monozygotic twin pair defined by schizophrenia. We've done single-cell RNA-seq to ask if the cells are making the same types of neurons, and in general they are, although there is an absence of patient-derived neurons in the fully mature neuron cluster. And we can quantify that and show that in the most mature neurons, clusters 4, 9, and 8, we are not getting enough patient-derived cells. The neurons from patients, whether they be 5' or 3', have fewer branches and less neuronal activity. And this question really is why. To what extent do these phenotypes reflect perturbations in few or many Neurexin-1 isoforms? Put another way, are neurons from patients firing at a reduced rate because one really critical Neurexin-1 isoform is missing, or broadly because all of the Neurexin-1 isoforms are decreased by 50%? And so that's exactly what we undertook. This was work led by a former PhD student in my lab, Aaron Flaherty, and a former postdoc, Shuja Zhu, in my collaborator, Gong Fang's lab. So here again, we have long-read sequencing of neurons from the 3' deletion carriers. The first thing that we can see is these orange isoforms. These are all significantly decreased in patient neurons relative to controls, and these gray isoforms that were not even detected in patients relative to controls, although I'll note they're the least abundantly expressed ones in the controls. And so overall, we have gross dysregulation decreased expression of many Neurexin 1 isoforms in the heterozygous carriers relative to controls. But something else was happening too. We have expression of 31 mutant isoforms in these patient neurons, isoforms that we never saw in control neurons and we never saw in the postmortem brain. And they're caused by splicing around the deletion from the heterozygous allele. And notably, they're the most abundantly expressed isoforms in these patient neurons. We wanted to know if they were functional. And so we cloned some of the wild type and mutant isoforms and overexpressed them in control and patient-derived neurons. If you overexpress any one of 4 different mutant isoforms in control neurons, you dramatically impair neuronal activity. If you overexpress wild type isoforms in these 5' carriers, the ones who don't express mutant isoforms, you increase and rescue neuronal activity. But these 3' carriers, the ones who express mutant isoforms, cannot be rescued simply by overexpressing wild type isoforms. And so we think 2 things are going wrong here. All the patients are affected by a loss of Neurexin 1 dose, and a subset of patients are affected by the additive effect of mutant isoform activity. Now, did we happen to find the only 2 Neurexin 1 carriers on the planet who make mutant isoforms? No, we don't think so. Diving deeper into the genetic studies of Neurexin 1 carriers at the time, our best estimates are about 1 in 5 Neurexin 1 heterozygous cases might be producing mutant isoforms that are translatable. After Aaron graduated, this project was picked up by Michael Fernando, a really fantastic PhD student in the lab. He wanted to really understand the cell type specific effects of these wild type and mutant isoforms. And so now he's moving into pure populations of glutamatergic and GABAergic neurons. The glutamatergic neurons are generated by NGN2 overexpression. He can dive in and by multi-sort array confirm this reduced activity of the glutamatergic neurons over development. And in GABAergic neurons, unexpectedly, he's actually seeing a hyperactivity of immature glutamatergic neurons during neurodevelopment. He can confirm this by electrophysiology. So here by ePhys, he's showing reduced frequency of EPSCs in both the 5' and 3' deletions. And in the GABAergic neurons, he's showing hyperactivity of GABAergic synapses. You can visualize these deficits here, and you can see not just that they're changing by cell type, but that they have differences in magnitude between the 5' and 3' donors. He really wanted to definitively show the impact of loss of function versus the mutant isoforms. And so to mimic the loss of function, here we're taking control neurons and knocking down wild type Neurexin 1 using a short hairpin RNA. And he can show that by the knockdowns, it's giving about 50% reduction in Neurexin 1 levels, and it recapitulates the 5' carrier phenotype, reducing activity, synaptic activity in the glutes, and increasing synaptic activity in the GABAs. So knockdown and increased synaptic activity, the blue line. To look at specifically at the mutant isoforms, now he's going to knock down mutant isoform activity in the 3' carriers to see to what extent that rescues the deficits and makes them look more like controls. He's got a very good knockdown by short hairpin RNA, and that knockdown here leads to increased frequency of synaptic activity in these mutant neurons. While the knockdown is not as robust in the GABAergic neurons, it is sufficient to reduce the hyperactivity of GABAergic neurons. Towards therapeutic interventions, we can imagine that you might actually need to do 2 things. In all Neurexin 1 carriers, you would need to increase Neurexin 1 expression, and then in a subset of carriers, you need to knock down mutant isoform activity as well. Our hypothesis, given that the estrogen receptor binds to the Neurexin 1 promoter, was that treatment with estradiol might increase Neurexin 1. And what we were able to show is that in glutamatergic neurons, but not GABAergic neurons, we were indeed able to increase Neurexin 1 using estradiol treatment, and the functional analysis is ongoing, but it does seem like estradiol treatment is increasing neuronal activity in patient neurons. To test a more therapeutic approach to knocking down Neurexin 1, we have antisense oligos designed. We're able to show that we can knock down mutant isoforms using these antisense oligos, but the functional work is ongoing. And so the idea here is that, we have this idea that one way of, altering genotype-phenotype relationships, especially in disorders like autism and schizophrenia might be to take advantage of sex hormone effects and that inherent sex bias of many brain disorders, where autism, for example, is strongly enriched in males. Estradiol might be a protective way of reducing the impact of autism-related mutations. Another environmental factor that we know impacts risk for psychiatric disorder is stress in the mother in second trimester. And so we also have been very interested in understanding how stress and inflammation impacts risk for neuropsychiatric disorders. And we'll add there's a second adolescent period where second hits induced by stress and drug abuse might also exacerbate phenotypes. So to look at the impact of stress on psychiatric risk genes, here we have stem cells derived from 39 donors. And again, we've made them into NGN2-induced glutamatergic neurons, and we're stressing them with low and high dose of the stress hormone H-cord. You can see that the more we stress neurons, the more their gene expression changes. The types of genes that change expression when we stress them are enriched for clusters of neurodevelopmental and immune response genes. But these 39 donors are actually all combat-exposed veterans from the VA recruited by Rachel Yehuda. 19 of them had PTSD and 20 had controls. And so if we break down this data from above by diagnosis, what you can see is that you see a stronger PTSD control effect at low stress than at high. So what we think this means is that neurons from patients with PTSD are hyper-responsive to stress. They're responding to stress at a lower dose than the control neurons. And those genes that are hyper-stress responsive at low dose are sufficient to distinguish neurons from cases and controls. The types of genes that are more stress responsive in cases are enriched for known PTSD risk genes that are coming out of post-mortem analysis and also genetic studies, but they're not specific for PTSD genes. You can see that we're broadly enriching not just for PTSD genes, but genes linked to autism, schizophrenia, epilepsy, and developmental disorders. And so we think that broadly, stress is dysregulating psychiatric risk gene expression in neurons. And so if sex hormones are perhaps ameliorating genotype-phenotype relationships, stress hormones might be making them worse. And so I'm going to segue now from talking about gene-environment interactions to talking about gene-gene interactions. How do risk variants sum? This is a long-standing project in the lab. It was started by Sukman Ho, my first PhD student. Here, Suk is showing that we can up and down regulate schizophrenia risk genes using CRISPR activation and inhibition, that we manipulate these risk genes, we enrich for downstream gene expression changes linked to synaptic function, and that by modulating common variant schizophrenia risk genes, we can actually see effects also at synaptic function by electrophysiology. Here, reciprocal perturbations of SNAP91 up and down lead to reciprocal synaptic deficits. But again, the question here is not if we can manipulate risk genes one at a time, it's what happens if we manipulate them in combination. And so here we've prioritized the top 4 schizophrenia GWAS genes based on the strength of the EQTL data in the postmortem. These were prioritized 7 or 8 years ago now. And so there were 4 genes with really robust evidence for genetically regulated gene expression in the brain linked to schizophrenia. We manipulated these 4 genes one at a time and did RNA-seq. And then we added the individual RNA-seq data sets to create an expected additive model. And in parallel, we actually did the experiment where we manipulated all 4 genes in the same well, and we asked how good our additive model was, with any differences between what we expected and observed being termed synergy or non-additive effects. And the model actually genome-wide was really good. 82% of the time, we could predict genome-wide gene expression changes based on the additive model. But 7% of genes were more down than expected. Those more down genes were enriched for the major neurotransmitter classes, and 11% of genes were more up than expected. Those genes that were more up were enriched for the rare and common variants linked to schizophrenia. And so what we're really saying here is that there is an emergent biology that you can only see by manipulating risk genes in combination that you cannot predict by manipulating risk genes one at a time. And so that if we want to understand the impact of dozens of risk variants in combination and how they impact psychiatric disorders, the only way to know that effect is to actually manipulate those risk genes in combination. And so that's what we've been doing. There's one caveat from this original study, which is those 4 genes we manipulated that were prioritized by the strength of the GWAS and EQTL data 3 of the 4 of them actually have roles in synaptic function. And so we weren't sure if this bias for synaptic function explained this non-additive effect. And so we've gone back more recently, now that there's a larger reference panel of the brain, and we've gone from PGC2 to PGC3, instead of having 4 genes to manipulate, there are now 25 potential candidate genes worth looking at. From those 25 genes, we pulled out the top 5 synaptic genes, the top 5 regulatory or epigenetic genes, and the top 5 multifunction genes, we're calling, that are neither synaptic nor regulatory and have no apparent relationship to each other. And so all 15 of these genes we're manipulating one at a time and also in combination. For the 5 synaptic genes, just like before, we see about 15% non-additive effects. For the regulatory genes, we likewise see about 15 to 20% non-additive effects, but the multifunction genes showed no non-additive effects. Digging in deeper, here you can really see these genes that did not respond as predicted by the additive model. And we tried to figure out what these genes had in common. And one of the surprising things that we found is, so these non-additive genes are identified based on the fact that if you manipulate the 5 synaptic genes alone and in combination, these gene effects are not predicted by the additive model. But if you manipulate these 5 synaptic genes one at a time and look for shared downstream effects where genes are changing the same direction for all 5 synaptic genes, there's about 1,000 convergent genes, and they are incredibly enriched for the non-additive effects. So we're finding these green genes by single gene perturbations and these blue genes by combinatorial, and they overlap. Likewise, looking at the regulatory genes, we again have very strong overlap of the convergent genes and the non-additive genes. Whereas for the multifunction, just as we had no non-additive genes identified, we have very, very few convergent genes. And so we wanted to dig into more about these convergent genes. What are they? Well, they're the rare and the common variants linked to schizophrenia risk. And they're really enriched for schizophrenia pathology. So we see them being enriched for glutamate receptor activity, GABA receptor binding, glutamatergic and GABAergic synaptic function. And so here, Michael, who started the work on the combinatorial analysis, has teamed up with Kayla, who's really focused on developing our scripts to look at convergence. To validate these convergence, we moved to a pooled screening format. So now we're including 10 of those original 15 genes and 7 additional genes. Kayla's illustrating here the convergent networks from those genes that have synaptic function and those genes that have epigenetic function. And the first thing that I want you to notice is that these convergent networks are actually distinct based on the function of the genes that are going into them. Whether we're looking at convergence by arrayed or pooled analysis, again, the convergent networks are enriched for schizophrenia risk genes. And we think they explain in aggregate a substantial portion of our sub-additive, non-additive effects. So again, to really help drive home what this looks like, if you have 10 risk genes and you manipulate them one at a time, you have different effects by gene that you can add to create an expected additive model. If you then do the experiment to ask how these 10 genes cause perturbations in aggregate, we generally see a less than full response to the combination. These individual gene perturbations have some elements in common. And those genes that are downstream of many of them, we're calling convergent targets. And those convergent targets are actually those ones that are explaining the difference between the expected additive model and the measured combinatorial model. And so the bigger picture here is that whether we're talking about gene-gene or gene-environment interactions, what we're trying to understand is genetic resilience, how to make sure each of us has, the most healthy outcomes. How do we minimize the effect of risk variance for disease and increase the effect for risk variance for health? And so with that, I'll stop. I've got to thank all the folks in the lab who contributed to all of this data, particularly for this example, Michael Deans, Michael Fernando, and Kayla Townsley, and Karina So, and our funding sources. And so here I am very happy to stop sharing and take questions from anyone in the audience. Thank you.
Thank you so much, Kristen. This was very wonderful. I while we wait for audience questions, I have a couple of questions to start. One is more technical oriented for many of our students who are new to iPSC world as this. How do we ensure especially when you're going from during the process of conversion, when you have one multipotent cell type to another multipotent cell type and onwards to a terminal deficient cell type, how do we ensure we have the cell type we think we have? I mean, markers obviously is the common way to go, but then many of the markers are not as restrictive to the cell type as it is commonly published. For example, neurons, people use beta-3 tubulin, but then beta-3 tubulin is also there in neural crest cells, which come way before neurons do. So in that sense, how can, in the daily course of things, we can ensure we've got the cell type we think we've got? And the second question is more broad conceptual, which is you talked about the combination of gene and environment. So modeling that interaction in a dish for example, a high genetic risk in combination with the estradiol, for example. How might we model that even for disorders that may not have a clear-cut gender bias, like a broader context of modeling gene environment interaction in the dish?
Yeah, okay. So I'll try to answer the first question first about knowing what cell type we have. I mean, this is incredibly important. There is a lot of variation in stem cell models. If we use the exact same protocol, but from two different donors, there will be some inherent variability in the yield and the maturity and the cell type composition of the cells that we make. And so yield being the percentage of cells that are what you want them to be, cell type composition also considering those cells that aren't what you want them to be. How much variability is there in that? So for example, if you have 80% neurons and 20% other cells, you know, if one person you have 20% astrocytes, but in one you have 10% astrocytes and 10% neural crest, or 1% of 20 different cell types, that variability in the non-right cells can also confound it. We have variability between people, and then there's stochastic variability. If you do the exact same cell line today and tomorrow and the next day, it will vary again. And so we do look at markers. They are imperfect. There's no single gene that we can really differentiate cell type. And so we actually have to account for this usually in our RNA-seq. It's where we're best able to adjust for this because we can take into account all of the best single cell data sets and impute the cell type composition within our RNA-seq. And so that's typically how we do it best. The second question about gene environment, I think you really have to know the biological or have a strong hypothesis about the biological effectors of the environment. So, right, like if the environment is a verbally abusive environment, it's not the sound itself that is causing the changes in the neurons, right? It's probably the stress hormones that are coming up. So you would be wanting to, know the actual effector that's acting on the neurons. This is actually really complicated during pregnancy. If we're talking about infection in the mother in the second trimester, was it the bacteria causing the infection in the mom that's ultimately impacting the fetus neurons? Or is the infection causing an increase in a cytokine? And does that cytokine cross the placental barrier? Or is it a new cytokine being expressed by the placenta? And does that cytokine in the fetus cross the blood-brain barrier? Or is there another upregulation of something else? And so for gene environment interactions, right now, I think the most important thing is to keep in mind the exact biological effector acting at the level of the cells in the brain. And that can be extremely challenging. I don't know if I fully answered your question, but I hope so.
No, you did. Thank you. Any other question for Professor Brennand?
I see something in the comments. Do sex hormones alter the expression of certain orexin 1 splice variants over the other? It's a fantastic question, and we don't know the answer to that yet.
Yep, people can raise hands and ask questions, I think, as well.
It's great that there's so many of you here. Rachel.
Hey, that that aspect of splice variance is really interesting and important. So when you study with respect to different conditions, do you see prevalence of any particular? I mean, would you predict? I mean, I'm sure you haven't done the study, but would you think certain splice variants become prevalent in different situations, and hence those splice variants could be the ones which are responsible, and later on maybe using it as a marker for diagnostics? Did you understand?
You're asking if changes in splice variants could be a biomarker, I think, right?
Yes. And will there be common splice variants, common set of splice variants with different conditions that you are studying, which would affect the whatever disease? I mean, like if you use, say, hormones, stress, in this thing, a common set of splice variants come up. Would you expect something?
I don't know if it's... I don't know if it's my internet, but I only heard about a third of your question, Rachel. So I'm going to guess. I know that you're asking about whether we can use splice variants as a biomarker and to what extent splicing might read out, I think, gene-environment interactions. I don't know the answer. I mean, biomarkers ideally would come from the blood and not from brain expression. And so we'll have to look to see, if we can detect these variants outside of the brain, or if we can predict their expression based on genotype. I'm very interested to know to what extent there's perturbations of splicing in idiopathic, right? Like non-Neurexin 1 deletion carriers. There are, in fact, some splicing variants that are common variants, right? That might impact Neurexin 1, even in those patients that don't have deletions. And so that's a really interesting question. But again, we don't know how Neurexin 1 changes by context. I do believe we've set up the samples, but nothing's back yet sequencing wise.
Yeah. I mean, I know that you haven't done these studies, but I was just trying to, initiate some thought processes. I mean,
I think it's very interesting.
Thank you. Let's not get.
Yeah. Akshay, you have a question?
Yeah, can you guys hear me?
Yes.
Now,
The question is, have you looked at any of these variations in schizophrenia or bipolar disorder having an additive effect on autophagy or lysosomal pathways? Because most anti-manic drugs have been proposed to contribute to autophagy, but they've not exactly been proven yet as like in neurodegenerative diseases. And typically, circadian rhythm disruption also regulates autophagy initiation. So, have you looked at any of those?
We have not looked at autophagy in around schizophrenia yet. We've got the assays set up for looking at neurodegeneration, but we haven't applied them to psychiatry yet. I think it's a really good idea.
Thank you. Any other questions for Professor Brennand?
Thank you all so much. I really enjoyed sharing our work with you today.
Thank you. Thank you so much for joining and making time for us. I think if there is no more questions, we will move on to our next speaker. Professor Andreassen. He is the professor of psychiatry from the University of Oslo and director of Norwegian Centre for Mental Disorders. His interest is in applying clinical, neurocognitive, and brain imaging phenotypes and molecular genetic tools to identify causes of mental disorders. He's one of the lead investigators of the Psychiatric Genomics Consortium, where he chairs the Bipolar Disorder Working Group and co-chairs the Alzheimer's Disease Working Group. His topic today is recent progress in psychiatric genetics through international collaboration. Professor Andreassen?
Thank you. Can you hear me?
Yes.
And can you see my screen?
Absolutely.
Thank you. So thank you for the invitation. I'm happy to present some of the recent progress in psychiatric genetics, and then focusing on the role of international collaboration and what that has enabled us in the field of mental illness genetics. And here are my conflict of interest. I will start by talking a bit about the polygenic landscape of mental illness and neuropsychiatric diseases. I think that's quite important frame shift in the field. Then see how this can be linked to brain-related traits, also large international studies, and then bring some examples of how we can move this discovery in imaging and genetics to see if we can get some help in the clinic. So I'm trying to focus on the big question of precision medicine, and I think this paper by Francis Collins and the head of the big All of Us program in the US formed Joshua Denny formed some interesting approaches, what is needed in the field now. And as you can see here, for this will affect the whole field of medicine, healthcare in the years to come. And as you see, what they have lined up here is routine genetics, big data, brain imaging, and also electronic health records. And I guess you guys in India is like in Norway, a large number of people have cell phones. So how this can be integrated in the research is quite important. And then also in phenomics, how your heart is functioning and everything on mobile registration. But then there are three last items here that's really important, that also large longitudinal cohorts are needed. And I will tell you a couple of the examples from the Nordic countries. Then we need trust, in people taking care of our data. And then also we need diversity. So most of the research I'm going to talk about today is mainly focused on European, US samples, and that's just not possible to do anymore. We need to make sure that the discoveries are present also outside of the US, European ancestry. But anyway, so all of this I like with this paper, but one thing I strongly dislike, they didn't mention mental illness at all, because I think that precision medicine is really important. All of these items are key for like doing more precision psychiatry, and everything here is important to build the field of precision medicine in psychiatry. And I mentioned also this, that we have this why is this so important in psychiatry? Because it's so heritable. Here are just the top of the list. You see there are really heritable, most mental disorders, and even depression, anxiety has pretty high heritability, but bipolar, schizophrenia, ADHD, autism is of course the highest. And this, due to technology, as you all know, it's now so easy to do large-scale genotyping, and at least on our hospital in Norway, or it's cheaper to genotype your DNA, then park at the hospital, and this will transform healthcare. And that is why the PGC, the Psychiatric Genomics Consortium, was established, because first of all, we have now these heritable phenotypes. It was very difficult to discover any genes on most psychiatric diseases. And then we have the technology, so we can do large-scale screening. And this has led to really a change in the discoveries in mental illness, psychiatric genetics. And you can go and look on this site. It's really well organized. It's an open, I'm a chair of the bipolar and also working on the co-chairing the Alzheimer, and it's open, everybody can join. It's all the data are published, you can download and everything. So I think it's a new way of organizing research. And this is also the only way to discover polygenic factors. I will come a bit back to that. Anyway, so it's been amazing progress most recent years, and everybody here, I guess, understand what is genome-wide association study. This is a Manhattan plot showing all variants in the genome, common variants, 2, 3, 4 million, and most of them have no significant association with the phenotype, here schizophrenia, but some, you see the skyscrapers, this is a log P scale, so the taller the skyscrapers, the more significant are the findings. And all over the genome, you can see effects here on association with schizophrenia. Came out last year, 17th of May. Bipolar disorder, 63 loci, same picture. We have now preliminary data presented this year at the World Congress last year. You could see this presentation is close to 300 loci now, coming on bioRxiv soon. Major depression just came out, a study building on many of the PGC findings. You now had 243 loci. And you see the picture is multiple loci across the genome. So it is really the polygenic architecture. And then ADHD, the same, not as powerful yet, but you see the same picture, many variants. And then even Alzheimer's disease, you can see here the old friend ApoE, huge skyscraper, of course, super significant. But then you can see there's a sort of under the big skyscraper, you can see all these smaller buildings arising. So there is a polygenic signal also in Alzheimer. And the most recent building on the PGC findings, there are now 75 variants. So it's mixed here between the polygenic signal and the ApoE. You can see it here. And also, education, as you all know, you can do GWAS, you can do an intelligence. These are also polygenic phenotypes. So this is the new kind of way of the new status in the field. We need to accept that these diseases are polygenic. And of course, now there's a large focus also on rare variants, doing sequencing, but that's still in the beginning. So I will focus today here on the polygenic signals. And it's also interesting, if these disease groups are so polygenic, what does it look like in brain-related traits, not only diseases? So if we look at the mental illness, a person in front of you with a disease, there could be all kind of different variants of the clinical manifestations. The people have sleep problems. If they have depression and schizophrenia, they could have appetite issues. Also, if they have other diseases, not only eating disorders. So for individual people, there is not like, oh, I have only sleep problem, nothing else. I only have cognitive problems, no problems with activity or mood. I only have delusion, nothing else. So there's a kind of mixture of the phenotypes that we need to also study more in detail. Then, of course, the comorbidity. There's a large overlap between depression and anxiety. You can see here, this is American data, severe mental illness and substance use disorder, like 30%. So there has then, or we think there should be some kind of overlap in the genetics also to explain this comorbidity. And then I will talk a bit more about this later, but also in brain imaging, there are large international efforts. And the NIGMA, we're also working closely, and I'm actively in the bipolar consortium of the NIGMA. And you can see here, bipolar, small effects, abnormalities across the whole cortex. Depression, less so, OCD, less so. Schizophrenia has the most abundant, and then alcohol use disorder, not so much. So, but there is not like one structure, one kind of cortical area that affecting one disease. So there seems to be this kind of pattern of overlap also on the phenotype, imaging phenotype. Then I just said that there's a large heritability as I showed earlier. So how can we then understand this? And as you know, there is a large genetic correlation, meaning coheritability. These diseases of mental disorders, they have genetic risk of one also influences the risk of another, and both ways. So it's more like a summary measure. So depression is highly correlated with ADHD, with anxiety, with bipolar. Schizophrenia seems to be associated with most other diseases. So there's a large degree of correlation, but some we don't find much. But there are other ways of doing the analysis. So I think what we, and this is what we've been working on now the last years, so the standard tools here doing the genetic correlation are based on the infinitesimal model, meaning all markers on the genome have an effect. And this is nothing wrong with the whole concept of twin studies and everything. Is you do this kind of correlation based on the more than 100 years old theories. But we have been developing something called the mixer package, where we do use a mixture model where you have null and null effects, more like a Bayesian language, as many of you know. So this is just an illustration that if you have all variants in the genome here and the numbers on the y-axis, effect sizes here, the standard tools are sort of saying that everything has an effect. But I think you all agree that fitting a model where you have the scales here are, their distributions of these have an effect on each side in a protective and increased risk, but most of the variants in the genome don't have an effect. So if you put that into your models, you can fit them better than this, everything has an effect. And then you can see now, usually with GWAS, you can detect variants that are beyond this 10 to the minus 8, the multiple testing on each side here. It's a very small proportion, but we're saying that, wow, maybe we can model the whole structure of these non-null effects. So then you can estimate, because we have so much data now, that we can estimate the proportion of the variants in the genome that has an effect on a given phenotype. And what also is difficult with the standard tools is to include opposite effect directions. So there is very little evidence that you everywhere in the genome has to have either protecting against another phenotype or increasing the risk. There could be regions of the genome where you have opposite effect directions. And this obviously here that, this is just negative effects here and positive effects, but they could be mixed across the genome. And this is something we've been working on in this method called mixer. And here, this is a way to illustrate that here are the effect sizes for depression, the blue ones. Most of them don't have an effect, they're very small, but then you can see each dot here is also a SNP common variant, and here are the effects on ADHD. And you can model them. You can now, with all the data, you have like millions of variants for each individual, and you have like hundreds of thousands of individuals. So this can really be used for mathematical modeling. And then you can model this overlap. And so what we estimated here is that for depression, there are like 14,000 in total. And just to illustrate, so you're now talking about this, what is the turquoise color here? That's the 14,000, but what I showed earlier on the Manhattan plots, that's the 247 that's discovered. But these are the estimated numbers. And the same with ADHD. We had 27 now that's discovered beyond the genetic, Conferone 10 to the minus 8. But there are estimated to be 5,000 in the polygenic signal of ADHD. So this is the number of the polygenicity that's estimated, for these two diseases. And then you can estimate the overlap. And this is quite significant. It's like 4,400 estimated across the genome with opposite, it could be same and opposite effect directions. So here you can see the same for schizophrenia, bipolar, large degree of overlap, just a little small piece here of the schizophrenia non-overlapping with the bipolar. And you can see schizophrenia and MD, schizophrenia and ADHD, large overlap. Depression and MD, you can see a large proportion of depression is not associated with the bipolar, but I guess this large proportion here is shared, and that's probably the mood, the depressive phase of bipolar. But here on the left hand is the section not associated with depression. That's probably the manic kind of genetics of bipolar disorder. And here is ADHD and bipolar that I showed earlier. That and you showed earlier with ADHD, and you can see here the same picture that ADHD is sort of part of the bipolar genome, and this fits with the notion that ADHD is often kind of developed when you're a child, and people when they get older they're diagnosed with the bipolar. The same picture with neuroticism, there's a phenotype, and also with subjective well-being. You can see there's a large degree of overlap. And then the general concept here is that the genetics of disorders of mental illness are overlapped also with personality traits, and especially here, so obvious, clear, positive correlation between neuroticism and the major depression. But then what about the brain? So you can use the same thing. If you combine the samples, you start to also identify the genetic factors associated with brain structure. And it's the same picture there. It's a lot of variants, and it's like for area, they're like 187, cortical thickness 50, and this is also driven by large-scale collaboration. This is 51,000 individuals, and all of them have these highly kind of complicated brain scans done all over the world. And this is thanks to the NIMHANS consortium that they've been able to collect these data. And then you can do, this is a free surfer, where you can estimate every little variant or region of the brain, everybody's talking about machine learning, artificial intelligence. This has been around for a long time. So this is just showing that if you can apply large-scale data and do this kind of data analysis tool that's now called machine learning, and find reliable estimates of the brain and the genes determining the thickness and area. Anyway, so what could there be any overlap here? So we've done the same with like schizophrenia here and brain imaging, just using this as a template. And could there be any kind of overlap? Because it's been hard if you do the genetic correlation work. You don't find much overlap with bipolar and schizophrenia and brain structure, even though it is these diseases are definitely based on non-functional brain signals. So how is that possible? So we say, okay, let's look at it with the mixed effect direction. So this is the data. You see, schizophrenia is more polygenic. It's like 10,000, but the surface area here it's like 2,000 variants, but most of them are inside of schizophrenia, even though the genetic correlation is like 0, because they have mixed effect directions. Some regions of the brain maybe have a larger surface area, some smaller, if you have tendency for schizophrenia. And the same with thickness. It's no significant genetic correlation. There's no overall signal. It's not like the thickness is thinner or smaller everywhere in the brain in people with schizophrenia. So there is this mixed effect direction that is driving the signal. Then you can do the same with bipolar disorder. It seems to be a bit lower, interesting here, lower overlap on the surface area, meaning maybe the surface area could be more of a neurodevelopmental trait. But anyway, there is also here clearly showing overlap, thickness and surface area, despite no significant genetic correlation. So this is not a risk factor or like a overall genomic correlation, but it's this mixture of effect and the polygenic signal that's driving this overlap, seen here with mixer. Then what can we use these findings for? Of course, you can move into biology. You can use enrichment tools, and you can find all kind of expression patterns, and that's, I think, highly available. But it's more into the future, because I think it is quite difficult to go from common variants into, wow, this is the biology. So there you think what we're doing now is trying to link it to sequencing studies to find, using the, like that we did in the most recent schizophrenia paper, that you find the genetic signal, common variants, to sort of select the genes, and then you can see if there are rare variants in those genes. So I would then like to finish with a few minutes about how we can apply the current common variants to clinically relevant models or tools. And that's highly important now for Alzheimer's disease. That's the best example so far, because you need to identify them before the disease gets too bad. And there are several, as I showed you, common variants now associated with Alzheimer's beyond APOE. And again, you can use this kind of supervised machine learning, because you have so much data now, and training and test models to fit the models and then use it in your prediction tools. But then you need to, even though the kind of GMOSs are great and everything, PGC, we have discovered a lot of stuff, but it's not so interesting to know that you will develop Alzheimer's. It's much more important to know when. When will you develop Alzheimer's disease? So that's why we have developed a method called polygenic hazard score. And then you can put the bins of different age of onset common variants into survival analysis. This is based on 17,000 AD cases and 37,000 controls. Controls then being last time seen healthy, because it's much more important if you're controlled that not have Alzheimer's when you're 95 years old than if you're 50. And then this here, age 60, all are healthy, no Alzheimer, and then 100 years old, we didn't have more data. So, and the worst quartile is the fourth here, the red. This is the real and the model data, and the blue is the least affected. 50% have survived, no Alzheimer here at 75 years old. 50% have developed disease with a worse kind of risk, but then with the least risk is closer to 95. So it's nearly 20 years age range for when you can predict age of onset. And also, if you then follow this, we didn't have more data here. So, but if you follow this line here, it seems to end up at like 115 years old. So everybody will develop Alzheimer when you live beyond 115 years, it looks like. And then you can plug this into, if you know the population prevalence, this is relative data, this is absolute risk, and you can see like 80 years old, population baseline is 1.4. The first percentile, only 1.3 will develop the disease, but in the highest is nearly 11. So it's a really, really large increase in the risk of developing disease if you combine ApoE and common variants. And this is important now, and with all the new drugs coming to the market, it's good to predict the lecanemab now, is although the previous one that were withdrawn, but now it's in the face of several new drugs coming to the market. And then you need to select the right person at the right time to give the drug. So here we have, again, combining imaging with the genetics, because we need to get as much information in the predictors as possible. Because if you come to the memory clinic, you need to know when to start the treatment, because if you wait too long, if the brain is too damaged, if you have too much memory loss, no drug can help you. Then the damage is so severe, so you can't repair it. So what we've done here is combine genetics with imaging to like a multimodal hazard score. And there are automatic programs now. We can calculate the whole range, not only the hippocampus here and that on the left-hand panel, but the whole basic ganglia, singular frontal hippocampus, occipital, other, in like a poly imaging hazard score. Because it's the same concept there. There are many small effects that are working together. It's not like one big thing. And you need to capture the whole variation. And then how can this work in real clinical setting? Well, you are sitting in a memory clinic, you receive a patient, I have memory loss. That's the complaint. Then you see the patient here at age 0, and then you follow them for 5 years, and then you can predict how many here all are healthy, and then you can predict over 5 years in the different risk bins of only having polygenic hazard score. So you can see those with low score here in blue, they have a very small chance, and the highest risk have a nearly 90% chance of developing Alzheimer after 5 years. But the odds ratio 20/80 is like 3.6. It's very borderline useful in the clinic. Okay, but at the baseline, when they come to the clinic, what else you do? You put them in the imaging scanner. And then you add this multi-imaging hazard score. And now the odds ratio is like 13. And you can see the pattern of these guys up here, very low risk after 5 years of developing Alzheimer. Down here are the ones that are much higher risk. But finally, you don't only do genetics and imaging, you also do cognitive testing at baseline. And if you add that information, then it looks like here. So then the odds ratio is 27. And the 3 groups on top, they don't need treatment. They don't need to be followed up. You can tell them here, you guys, you are in this kind of part of the risk. You will not, or highly unlikely, develop Alzheimer. We don't need to worry, and you don't need these drugs. But here, this is the region of people that will develop and when they will develop. So you need to start before there's too much damage done. So this can be used to stratify treatment, the new drugs coming in. And this is also quite important, even though we have now new drugs, but you can also use this to reduce the noise in the data in a clinical trial. So, if you, I got some updates now. I want to quit this, sorry. So if you do a clinical trial with everyone in the street, you have, that's like 100%, then you need 100 patients. If you do age and do, then you need 70% of kind of the man in the street approach. If you then add polygenic hazard score, you can reduce the sample by 50%. Then you can see if you add imaging, it goes down to 35% of the numbers needed in the clinical trial. And that's super important for reducing the cost of running clinical trials. Then you can also see that you get not that much improvement in doing the cognitive testing. So roughly 1/3 is what you get out of this. And you can save 2/3 of the money on recruitment and follow-up in a clinical trial. And then if you want to put this into action, then you need to also put this into the healthcare system. And this is, you can have then a kind of cockpit for the doctor where you can add risk reports and cognitive tests and imaging into a physician portal and then develop integrated risk report, because this is needed if you want to move into precision medicine. And this is also what we do in the Nordic region. We have like 25 million people there, not so many as you guys, but we have registry data, biobanks for many, so we can sort of use real world data to train the models, and then we can have specific clinical sets to test them. And this is what we are working on now quite extensively. Anyway, I'm getting to the end here. So we need international collaboration to discover these common variants, both brain imaging and genetics. There are a lot of genetic variants associated with neuropsychiatric traits and disorders. And there is this complex pattern of shared genetic architecture that we then can use in our multimodal data to improve prediction to be ready for precision psychiatry. So the next frontiers now is trans-ancestry data and using also longitudinal data. We need to understand the timing of events here. So thanks a lot. This is people at my center, or all the people involved participating in the studies, especially for the PGC and Enigma collaborations. Thank you.
Thank you so much, Professor Andreassen. Are there any questions?
Okay, I had a question, if I may. So, in your analysis that you showed, because you do both the dementia and the bipolar work, do you think there is a lot of correlation between genetic correlation between neurodegenerative conditions and, let's say, bipolar disorder or schizophrenia, in terms of the larger studies that you have done?
It's an empirical question. We've done it. We've seen it. There is, I think, from the phenotype epidemiology studies, you have a twice risk of Alzheimer's if you have a bipolar disorder, unfortunately. So there's some overlap with some, and we have done genetic studies to see that there is an overlap in polygenic risk factors between bipolar and Alzheimer's. Yes. But now it has been a bit difficult to fit these models because Alzheimer has this very strong signal in the ApoE region, and then the polygenic signals. But if we find a way to separate chromosome 19 and then model it separately, and then we get a better handle now on Alzheimer genomics also. And then I think that would be quite interesting to understand how and why people develop more Alzheimer if you have also other mental illnesses.
People can raise their hand and ask questions. Well, if not, I have another question. So with the a lot of international collaboration across ancestral diversities now, which was not there in the past, do you think there is scope for meta-analysis, or do you think there is scope for replication of studies from one ancestry to the other? Which do you think would be more, in a way, advantageous? Because the sample size, for example, in an Indian sample or a South Asian sample is much lower than a European sample, and it will take a long time to catch up. So what would be your take on how to approach this? Would it be replication, meta-analysis, or something else?
I have no strong opinions on that. I think there is somewhat, it's made a bit too complicated, I think, with the trans-ancestry. I think it's the LD structure is hitting the real biology. So if you get the way you can do this kind of local ancestry things, and I think then what we have seen. So in our most recent, is not published yet, but in our most recent bipolar disorder study, we found a lot of similarities across ancestries. So of course, we didn't find a real it was harder to find like an East Asian specific genomic, what do you call it, pattern. But why should there be other genetic factors driving the brain structure or something in India than in Europe? It's more like the LD structure is making them harder to find in a way. But then if you look at on the other side, when we added the different ancestries in the fine mapping analysis, that improved the way of identify, was easier to find the underlying kind of genetic signal that sort of captured by the layer of common variants, because the common variants are, of course, much more kind of affected by the different LD structures in the data. So I think this is, I would just use it whatever is possible, be opportunistic, I think it's hard to predict these things, yes, but I don't think it is, this is so different and everything. There are now tools that you can use it. And so we found, at least for bipolar, just adding everything into a big mega, that was the most or meta, I think. Well, we used both, kind of partly mega, partly meta. That was the most fruitful way of getting kind of knowledge out of the data.
Right. Perfect. Thank you. Okay, Bharat has a question. Why don't you just unmute and ask?
Yeah. Thanks, Ole. I think it's a fantastic presentation. I had a question on when you add imaging to the model with genetics and age, the polygenic and the age, you get a significant bump. What would happen if you add cognitive in the next step instead of imaging? Because imaging would be cost prohibitive in some instances. So if you only add the cognitive tests to the model, what would be the improvement? Let's say it was 13 to almost 27 odds ratio. What would happen in the reverse scenario?
Very good question. So I think we in the paper we did that. It just came out. Ria said that we're doing it with Anders Dale, and it's I think it was a bit lower, but not that much. So you can get very long way genetics and cognition. But then again, what the conceptual thinking here is that if you want to capture people before they get the disease, then both imaging and cognition is, a sign of neurodegeneration. You see what I'm saying? So I think that's if it could be that, of course, then you can track it early on in the disease. But I think what is the problem is that if you diagnose the disease based on symptoms, then it's very hard. It is kind of this catch-22. It's very hard to do kind of disease preventive treatments. So, and also if you do it after onset, then you need to start so early. So the symptoms are so diffuse. So it's hard to do the diagnostics. So that's, I think, genetics is really important to have in, because that, of course, is the same whenever the genes are the same, the variants are the same. So then we're also now working on adding biomarkers. There seem to be some indications that some of these new biomarkers for Alzheimer are also very early onset. So the more we can do on the kind of finding predictive markers that can be more specific of the onset, that is what is possible. But I thought you had a lot of imaging scanners around, don't you? Or is it, I know in the US it's nearly more expensive to do cognitive testing, because there you need to have psychologists and doing it and everything. So I think, yeah, these things are, but of course it's easy to do this cognitive testing. Yeah.
Thank you. Sushil Kumar Joseph, would you like to ask your question?
Hello, am I audible?
Yes.
So my question is, first of all, it was a wonderful talk, sir. So my question is, can we add socio-economic status and environmental pollution and other factors into this complex module to predict? Because we know AD is multifactorial. It has a complex interaction between environment and biology.
Then that's an excellent question. So give me the data, we'll do it immediately. So if you had the, the problem is, when we really can get large samples, that's case controls, that's it, yes, no diagnosis. But what we are working on now is to use the large biobanks and registries like in Norway. We have 400,000 people, all genotyped, followed over time. We have exactly this thing, what you're talking about. Could it be head trauma? There could be hearing loss. Could be high blood pressure, alcohol, smoking, all the environmental factors that are known risk factors for Alzheimer. And that explains quite a lot. I think 35% now are modifiable. And the biggest modifiable risk factor now in the US, that's obesity. So but you don't have this data, but if that's why I was talking about the longitudinal. So if the obesity started earlier and head trauma, when it happened, then you can start adding those things to the model. And I super interesting if you have some ideas also to kind of how this can be integrated. So I think what is needed, what we are sort of working on now is to find that like the normal trajectory, if we have repeated data, and then you can see, wow, here we start dropping because you had this kind of environmental or whatever, your genotypes say, oh, you go, this should be your trajectory. But then if you have, oh, something happened here, and it goes down, then something is going on. And this is the concept. We need to have this longitudinal data. Was that what you thought about?
Thank you, Ole. I think there are no further questions, so I think we'll move on. So with this, we come to the end of this webinar, which is probably the first in the series of seminars for the Center of Brain and Mind. We thank eLife for the Ben Barres Award for Dr. Ritika. We thank all the eminent speakers who have spent a lot of time with us, all the participants who have been very active and have been asking questions, all our funding agencies, the Rohini Nilekani Philanthropies, the Government of India funding through the DBT and the India Alliance, and our team here, the IT team, including Kadambari, Kubendran, and others who have been very helpful, the NIMHANS Digital Academy. Thank you so much. Thank you.
Thank you.
Thanks.